tumor necrosis factor receptor superfamily. Member 4, also known as CD134 and OX40, is a member of the TNFR superfamily of receptors which is not constitutively expressed on resting Norve T cells, unlike CD28. OX40 is a secondary co-stimulatory immune checkpoint molecule, expressed after 24 to 72 hours following activation. Its ligand, OX40L, is also not expressed on resting antigen-presenting cells, but is following their activation. Expression of OX40 is dependent on full activation of the T cell. Without CD28, expression of OX40 is delayed and of fourfold lower levels. Function: OX40 has no effect on the proliferative abilities of CD4+ cells for the first 3 days, however after this time proliferation begins to slow and cells die at a greater rate, due to an inability to maintain a high level of PKB activity and expression of BCL2, BCLXL and survivin. OX40L binds to OX40 receptors on T cells, preventing them from dying and subsequently increasing cytokine production. OX40 has a critical role in the maintenance of an immune response beyond the first few days and onwards to a memory response due to its ability to enhance survival. OX40 also plays a crucial role in both Th1 and Th2-mediated reactions in vivo. OX40 binds TRAF2, 3 and 5 as well as PI3K by an unknown mechanism. TRAF2 is required for survival via NFIB and memory cell generation whereas TRAF5 seems to have a more negative or modulatory role, as knockouts have higher levels of cytokines and are more susceptible to Th2-mediated inflammation. TRAF3 may play a critical role in OX40-mediated signal transduction. CTLA-4 is downregulated following OX40 engagement in vivo and the OX40-specific TRAF3D end effect was partially overcome by CTLA-4 blockade in vivo. TRAF3 may be linked to OX40-mediated memory T-cell expansion and survival, and point to the downregulation of CTLA-4 as a possible control element to enhance early T-cell expansion through OX40 signaling. Clinical significance: OX40 has been implicated in the pathologic cytokine storm associated with certain viral infections, including the H5N1 bird flu. An artificially created biologic fusion protein, OX40 immunoglobulin, prevents OX40 from reaching the T-cell receptors, thus reducing the T-cell response. Experiments in mice have demonstrated that OX40 can reduce the symptoms associated with the cytokine storm while allowing the immune system to fight off the virus successfully. Interactions: CD134 has been shown to interact with TRAF5 and TRAF2. References. Further reading